Okay, so today's lesson is about tissue fluid. So tissue fluid basically is the fluid that bathes the cells. Between the capillaries and the cells, there is this tissue fluid. And what it does is supplies glucose, amino acids, salts, and oxygen to the tissues, and it receives carbon dioxide from the tissues. So as I said before, it is the solution that bathes the cells, where materials are exchanged between blood and cells, and it is formed by blood plasma. And the composition of it is controlled by homeostatic mechanisms, so it creates a constant environment for the cells. So key things are, it bathes cells, it supplies glucose, amino acids, salts and oxygen to the tissue cells, and it receives carbon dioxide from the tissues. So how tissue fluid is formed is as the blood flows from the arteries to the arterioles to the capillaries, those vessels are getting smaller and smaller and smaller. And because of that, because you're trying to force a certain amount of blood through smaller and smaller vessels, what you actually do is create a really high hydrostatic pressure. So hydro means water, so you're trying to get a lot of liquid through a small vessel, so you create a high hydrostatic pressure at the arterial end of the capillary. So if you look at this diagram here, this is the arterial end, where the blood will come from the arteries to the arteriole into the capillary, and this is the venule end, sometimes called the venous end, but we'll call it the venule end. So here you can see that the green arrow is very big because that represents the large hydrostatic pressure which is trying to force the plasma out of the capillaries. So yeah, this large hydrostatic pressure forces the tissue fluid out of the blood plasma. But that outward pressure is opposed by two smaller forces. So you've got a small hydrostatic pressure of the existing tissue fluid that is around the cells, which is opposing the hydrostatic pressure. And you've also got the fact that there is a lower water potential of blood because of the plasma proteins that are too big to fit through. The plasma proteins lower the water potential, so it tries to pull the water back into the blood. But because of that large hydrostatic pressure, the overall movement of fluid is actually out of the capillary into the tissue fluid. So key point here, because of the vessels getting smaller from arteries to arterioles to capillaries, and you're trying to get a volume of blood through those, what that does is it creates a high hydrostatic pressure, which is this green arrow here. But you have two small opposing forces. So you've got the small hydrostatic pressure created by the existing tissue fluid that's already there. So that's that small green arrow. And you've also got the fact that the plasma proteins in the blood lowers the water potential. So that's kind of trying to pull the water back. But because this hydrostatic pressure is so large, the overall movement, even though you've got these two opposing forces, is out of the capillary into the tissue fluid. And that forcing of the liquid from the capillary into the tissue fluid, because you're forcing it or filtering it under pressure, it's called ultrafiltration, because you're trying to get the liquid out of these small gaps. So filtration under pressure, it's called ultrafiltration. So the large hydrostatic pressure creates an overall pressure that pushes the liquid out of the capillary into the tissue fluid, but the proteins are too big to fit through these tiny gaps, so the proteins will stay in the capillary. So the filtration of the smaller liquid under pressure is called ultrafiltration. So then when it gets to the other side, the venule end, most of the tissue fluid is returned to the capillaries. And the reason that it does get reabsorbed is because of the fact that a lot of the liquid is being squeezed out at this end. There's now less fluid in this end. So what you've been left with is actually a lot of proteins which are lowering the water potential. So the water potential gradient is going to be pulling, wanting to get water back into the capillaries, because all the liquid is being squeezed out at the arterial end. Apart from the big proteins, all you're left with at this end is the large proteins which lower the water potential. So those osmotic forces are going to try and pull the water back into the capillaries. And also, just the fact that you've got less liquid in the venule end now means that there is a bit of hydrostatic pressure, which is kind of, because you've got less liquid in the capillaries, that there isn't that push from the hydrostatic pressure anymore. It's largely reduced. So you can see the green arrow at this side is a lot bigger than the green arrow at this side, because it's all being forced out at this end, the liquid. There's hardly any liquid left in at this end, so the hydrostatic pressure is largely reduced. So what you end up with is, because of the osmotic pressure, because of the proteins lowering the water potential, then the liquid re-enters because of the overall flow, because of the osmotic pressure is higher than the opposing flow, the opposing force of hydrostatic pressure. So the liquid will be reabsorbed. So key points: the hydrostatic pressure outwards from the capillaries is largely reduced because of the liquid is being forced out the arterial, and there's hardly any liquid left in here. And the larger force is because you've got the proteins, just the proteins left in the venule end that are large, they will lower the water potential. So you've got an osmotic pressure which is pulling the water back in, which is higher than the hydrostatic pressure outwards. So the overall movement of tissue fluid is back into the capillary. So the tissue fluid isn't really, by this point, a great environment, because it's lost a lot of the oxygen to the cells. It's gained a lot of carbon dioxide and waste. So some of it is actually carried, some of the tissue fluid, because you don't just want it to stay there, is carried back by the lymphatic system. And the lymphatic system is a system of vessels that start in the tissues and they get larger and larger, and then the larger vessels drain the contents back into the bloodstream via two ducts near the heart. So that means you can get it to the lungs to get rid of the carbon dioxide a lot quicker, because the lymphatic system is going to take that tissue fluid with the waste and the carbon dioxide straight back to the heart. So the movement of lymph through the lymphatic system, the thing that drives this movement, is the hydrostatic pressure of the tissue fluid that's left in the capillaries. That'll create a bit of pressure, which is going to kind of move the lymph through the system. But there's also contraction of muscles around the lymph vessels, which basically squeeze the lymph towards the heart, a bit like peristalsis in the esophagus. There's like smooth muscles which squeeze the lymph vessels, so it squeezes the lymph back towards the heart. But there's also valves which ensure that the lymph goes in one direction, there's no backflow of lymph. So there's valves to ensure that the fluid moves towards the heart. Okay, so this is just a diagram to show like the overall flow. So blood plasma, initially it's the liquid part is filtered out by ultrafiltration. Most of the tissue fluid will be reabsorbed into the capillaries at the venule end. However, whatever's left will have a lot of carbon dioxide and waste, so that will drain through the lymph, through the lymphatic vessels, through the help of the osmotic pressure of the remaining tissue fluid, and also the squeezing of the lymph vessels and the valves to stop going their own way. So that squeezing will ensure that the lymph returns to the heart vessels that drain into the heart. Okay, so an overall diagram just to explain tissue fluids formation. So as you go from the arteries to arterioles to capillaries, the vessels get smaller and smaller, and that creates a large hydrostatic pressure at the arterial end of the capillary. There are two opposing forces. There is the osmotic pressure of the proteins, which are trying to pull the water back in, the fluid back in, and also a slight hydrostatic pressure from the existing tissue fluid that's already there. However, these two pressures are a lot less in comparison to the large hydrostatic pressure. So overall, the movement of the liquid goes out of the arterial end of the capillary into the tissue fluid. And that is known as ultrafiltration because the liquid is filtered through tiny little gaps, so the proteins are left behind because they're too big. At the venule end, the hydrostatic pressure has largely reduced because a lot of the liquid has been forced out at the arterial end, so you haven't got much liquid in this end, so it's a lot, the hydrostatic pressure is reduced. But however, because there's hardly any liquid but a lot of proteins in the capillaries, there is a large osmotic pressure which is pulling the water back in because you've got a lower water potential here. So that osmotic pressure is higher than the hydrostatic pressure the other way. So the overall flow of tissue fluid is back into the venule end of the capillary from the tissue fluid. So most of the tissue fluid gets reabsorbed this way. Any that is left will drain through the lymphatic system back into two lymph vessels which drain back into the heart. And that is summary of tissue fluid.